Well, I'd like to thank the organisers for the opportunity to talk to you today about some of the work that's been going on in my lab, and in particular some of the work that we've been doing looking at the regulation of two-pore domain potassium channels by mechanical stretch, and so their pressure sensitivity. So mechanosensitive ion channels underlie several of the key sensations that allow us to interact with and perceive our environment. In particular, the touch sensation and, of course, hearing. And they do this primarily through their ability to turn changes in pressure and, in particular, changes in membrane tension into changes in electrical signaling. But despite these important roles and their role in many other fundamental processes in the human body, we know alarmingly little about the molecular mechanisms that underlie mechanosensitivity of ion channels, and that's really what we have been interested in most recently. We work on my lab works primarily on the two-pore domain, or KCNK family of potassium channels. There are 15 members of this family expressed in many diverse tissues where they control a wide variety of different physiological processes. Now, the textbooks tell you that these are sort of relatively boring leak conductances, just generally involved in control of the resting membrane potential. But we now know from studies in particular of the TREC and TRARC channels, here, this particular subfamily, that these particular K2P channels exhibit a complex polymodal form of regulation. They can be regulated by many different factors, including temperature, voltage, acid, intracellular and extracellular, and as well as a wide variety of compounds and other signaling pathways, and also pressure or membrane stretch. So this enables them to integrate a wide variety of different signaling pathways into changes in electrical activity. Well, the one I'm going to talk to you most today about is this stretch sensitivity, or their mechanosensitivity. The TREC2 channels the TREC and TRARC channels are all intrinsically mechanosensitive. We know this because we can purify the proteins, reconstitute them into lipid bilayers, and change the pressure across the membrane to stretch the bilayer, and you can activate channel activity in these directly in these purified proteins. So they don't require being tethered to the cytoskeleton for this mechanosensitivity. It's an intrinsic property of the protein. And so we thought, well, maybe this is a good model system to try and understand what the structural basis of mechanosensitivity is in these channels. And we're very lucky in Oxford to be able to interact with Liz Carpenter, who heads the Membrane Protein Structural Biology Unit at the Structural Genomics Consortium. And in collaboration with Liz, we were able to solve the structure of a human TREC2 channel in just not just one conformation, but actually two conformations. So they look like the other K2P structures which have been solved. So they've got the main part, which looks like a classic potassium channel. They have this funny little cap domain on the top here. We don't really know what that's for yet. And these structures may look relatively similar, but this little morph here, if it's going to, it did play earlier. It's not going. Let's go back and see if it'll play. No. Anyway, there's some other pictures of it in a minute. It moves up and down to exaggerate the difference between these two states here. And most of the movement occurs in the lower half of the protein here. Yes, there's actually this one's moving. So this exaggerates the difference between those two states. So you can see where most of the movement between those two conformations is likely to be in this lower part. So this immediately proposed a possible mechanism by which these channels might respond to membrane stretch in accordance with what's called the force from lipid principle of mechanosensitivity, which predicts that a mechanosensitive channel will expand its cross-sectional area as you stretch the membrane and increase the area per lipid as you stretch the membrane. So this model of moving from what we call the down to the up conformation here seemed to sort of be a very attractive idea for how they might respond to membrane stretch. But of course, this is based upon simply the comparison of two relatively static X-ray crystal structures, and we wanted a more dynamic picture of what's actually might be going on. It was also rather further complicated by the fact that there were two contradictory studies published about how the TRARC channel, which is very, very similar in structure, might respond. There was one from Rod McKinnon's group that suggested that the channel might move from the down to the up conformation, but involve a lipid block within the pore. And there was another paper at the same time from Dan Miner's group, which suggested that mechanoactivation involved movement from the up to the down conformation. So we thought, well, what's happening in the TREC2 channel? So we decided to do some molecular dynamics simulations. And so for this, we took our structure, we embedded it in our simulation box in a lipid bilayer with some salt and water, and then we changed the shape of the box so that you increase the XY plane here, the area increases, so the area per lipid increases in this direction to sort of mimic stretch in the membrane. And then we quantified how the structure changed. And to do this, we compared basically you overlay the transmembrane helices and calculate something that we call the root mean squared deviation. So how much the alpha carbons in the backbone of those helices deviate. And if we compare this starting structure against the down state, and when you stretch it, it moves away from the down state and it moves towards the up state. So we can demonstrate that the application of symmetrical lateral force here does actually cause the channel to move from the down to the up state in response to increased membrane tension. It also causes, this is the plot of the cross-sectional area, the change in cross-sectional area when you stretch the protein, and you see a big increase in the cross-sectional area in the lower half of the protein. And so many of the slides that you'll see here, the zero value here represents the middle of the bilayer. But you might think that actually you embed any membrane protein structure into a bilayer and you apply these, you stretch the membrane, but perhaps every membrane protein would just sort of move apart when you stretch it. But we actually have the perfect control because we have the crystal structure of the non-mechanosensitive TWIC channel. It's another K2P channel. And if we repeat exactly the same protocols with this channel, we see no change in its structure when we apply even the strongest lateral forces to this particular protein. So this movement does appear to be specific to these mechanosensitive K2P channels. So where are the forces coming from? Well, all membrane proteins are subject to some quite extreme forces when they're embedded within the bilayer. So you can calculate what's called the lateral pressure profile across in this case, a pure lipid bilayer. Within the core of the bilayer and between the head groups, there are some compressive or repulsive forces between the head groups and also between the acyl chains here. They give you these positive forces, pressures in the bilayer. But in particular, at the interface between the head group and the acyl chains, you get these very large tensive forces here within the bilayer. So this is very typical of a pressure profile across a typical bilayer. And this changes when you stretch it. So you see a reduction, as the area per lipid increases here, you see a reduction in these positive pressures, particularly within the core of the bilayer. Now, the membrane thins very slightly, and the tension at these peaks here also increases very slightly. And so it's likely that the protein is responding to this change in this pressure profile as you stretch the membrane. So we seem to have this intuitively attractive model for how the channel moves from the down to the up configuration, perhaps like this, when you stretch the membrane. But this is a very asymmetric process. Perhaps not surprisingly, I mean, you don't want the top part of the protein to expand a great deal because this is where the selectivity filter is. You don't want this part to expand. These channels retain their potassium selectivity when you stretch them. So the top part here includes the selectivity filter, so perhaps not surprisingly. This doesn't change. The major structural changes are in this lower part. So you can really consider it as rather like a sort of an inverted funnel shape here. It sits in the membrane and then becomes slightly more symmetrical when you stretch the membrane. But we thought, well, if this movement is driven by changes in membrane tension and this change in this pressure profile, then can we drive movement from the down to the up conformation by simply increasing tension in just the inner leaflet of the bilayer and not the outer leaflet? So, well, how can you actually do that? Well, first of all, we tried a simulation approach. Can we increase tension in just the inner leaflet? And to do this, in our simulation box, we kept the size of the box constant, and we simply deleted lipids from either the inner leaflet or the outer leaflet to mimic this sort of increase in area per lipid in the bilayer. And this is the appropriate change in the pressure profile in the membrane. So, of course, you see an asymmetric change in the pressure profile. This shows removal of lipids from the inner leaflet. If you did it from the outer leaflet, it would simply be inverted. And so you could see the increase in tension here and the reduction in the positive forces within the core of the bilayer. So what happens if we quantify the structural change? Well, if we remove lipids from just the outer leaflet, then the structure. So this is comparing the transmembrane helices. There's no real change during our simulation. If we change the tension in the outer leaflet, but if we remove them from the inner leaflet, then the structure quickly begins to move towards the up state. So it seems that the channel can respond to an asymmetric change in the pressure profile, or asymmetric membrane tension. So when will this type of stretch occur, and why is it of any importance? Well, not all mechanosensitive ion channels are the same. They're not all going to respond to just symmetrical membrane tension. When we interact with our environment, when cells get damaged or you touch a surface, the membrane will get twisted in many, many different ways. And so the changes in the pressure profile are going to be quite complex in a membrane. And perhaps you don't want all mechanosensitive channels to respond to exactly the same stimulus. If you have a diversity that respond to different types of stimulus, then that will be important. So this introduces the concept of membrane curvature. Because actually, when we stretch the membrane in our recording configurations, you will induce a certain amount of curvature. And of course, the overall tension in the bilayer will increase whether you apply negative tension or positive tension. You'll have an overall increase in tension. But of course, because this structure is asymmetric and the change in cross-sectional area is asymmetric, then it should be able to detect the difference between these two types of pressure changes. So this introduces the concept of what's called shape sensitivity in membrane proteins, and the concept of membrane curvature, or local membrane curvature, and membrane torque. So if you have a protein that undergoes an asymmetrical change in its structure when you stretch, or between two different conformations, then it should respond differently to different changes in the pressure profile. So can we actually detect this functionally? We've shown we can do this structurally, but does the protein functionally respond to differences in asymmetric membrane tension? So is pressure activation asymmetric? Unfortunately, there's some conflicting data in the literature. Most of the studies say that the channels respond to just negative pressure and not positive pressure, but there's some that say they respond equally to both positive and negative pressure. And the literature is really quite confusing depending upon which paper you want to read. And this is probably a result of the fact that interpretation of pipette-based recordings of mechanosensitivity are incredibly complex and quite challenging due to the shape of your classical omega shape of the patch in the pipette, the fact that you've got extreme membrane curvature in some places, and also the difference in the adhesion energies between the bilayers and the glass. And so it can be very, very complicated to try and interpret mechanosensitivity, particularly asymmetric mechanosensitivity using pipette-based recordings. So we thought, well, can we use a different type of system? So for this, we thought maybe we can use a simple, reconstituted system in a defined planar lipid bilayer, because we can purify a protein, and I showed you earlier, it does respond to changes in pressure. So for this, we use the nanion Porter patch and their chips, and we reconstituted the protein into GUVs. And this is an image of one of the holes in one of these chips, and this is an image of some of the GUVs, labeled GUVs, over the surface of one of these holes. And there's another little picture here showing how those GUVs burst onto the surface of these chips. So using this system, we decided to try and look at the asymmetric, to look at the pressure sensitivity. But of course, we ran into an immediate problem in that the protein can incorporate randomly into the bilayer. So an attempt to do this macroscopically is essentially impossible. And actually, if we look at macroscopic currents, we see equal responses to positive and negative pressure. So I thought, well, can we actually do this at the single channel level? Let's give a really difficult project to the postdoc. So Michael Clausen was very patient, and in the bilayer, luckily Trek has an asymmetric conductance in the bilayer. It has a slightly higher open probability at positive potentials, and it has a much larger conductance of about, we use about sort of 200 millimolar potassium in these recordings. So it has a conductance of about almost 300 picosiemens under the conditions that we study. And we can actually monitor the orientation of the channels in the bilayer using side-specific blockers. So quaternary ammonium ions, such as tetrapentyl ammonium, will block the channel only from the inside. So we can define what we call, what we would define as the inside-out configuration, and we can use ruthenium red, which binds just below the cap region here, as an extracellular blocker to define that what we're looking at, that we really do know the orientation of the channels in the bilayer, in addition to just using their different conductances here. So what actually happens when we apply asymmetric stretch? So in this case, we've got single channels in the bilayers, and in the inside-out configuration, here we do in fact see a preferential activation by just negative pressure. And if we have a channel in the other orientation, then we see preferential activation by positive pressure, which would be the equivalent of negative pressure if the channel was oriented the other way. But this is just a cartoon here. There's bound to be some kind of complex geometry, and certainly this is not to scale. There's bound to be some kind of complex geometry here. Only one of the leaflets adheres to the bilayer. Perhaps this is an artifact of our recording system. So we thought, well, an important control might be to take a situation where you've got two channels in the same bilayer, one in one orientation and the other in the other orientation, and apply positive pressure, negative pressure or positive pressure, and see if you can get simultaneous inverse regulation of those two channels. And so that's but that type of analysis becomes quite complicated because, of course, once you've got more than one channel in the pipette in the bilayer, then the conductances can begin to superimpose. They're relatively different, so we can begin to you can begin to tease them out. But my postdoc Michael wrote a little bit of a MATLAB script to try and differentiate. With up to 3 channels in the bilayer, we can actually tease out which conductances we're actually looking at. And so what happens when you have that situation? Well, so this is sort of simultaneous inverse regulation of one oppositely oriented channels. When you go to negative pressures, the inside-out channels activate, but the outside-out ones don't. And if you go to positive pressures, then the outside-out ones activate and the inside-out ones don't. And of course, this is this would be the equivalent of negative pressure for this for the channel oriented the other way. So these channels do appear to exhibit a functionally asymmetric response to membrane tension. Another interesting point, because we can do rather nice single channel recordings, when you study these channels expressed in cell lines, unfortunately there's some alternative translation of the alternative translation initiation sites, and the N-terminus can vary, and you can get different size conductances. Single channel analysis is actually quite difficult with heterologously expressed Trek tube channels, but our purified channels exhibit some very well-behaved single channel behavior. We thought, well, maybe we can actually try and understand what stretch activation is doing to the single channel kinetics. So if we look at, the channel seems to exhibit a single open time and three distinct close times. So what happens when you activate the channels? Well, you see a very selective reduction in the long close state, as you can almost see very clearly in these single channel recordings. So just to summarize here, the channel undergoes an asymmetric change in its structure and expansion in the lower area when you in response to membrane stretch. And we think that this asymmetry is going to be important for allowing different types of mechanosensitive channels to fine-tune cellular responses to different types of different and diverse mechanical stimuli. And that some of the principles we've uncovered here may apply to other types of ion channels. We really want them all to be different and not to be the same. And so I'll just skip through this. And this is the down to up motion as the channel activates, that the local environment around the protein and this changes in local curvature and this pressure profile asymmetry are particularly important. And we think that actually maybe there are some advantages to planar bilayer recordings over traditional pipette-based recordings for these specific types of experiments. And of course, none of this would have been possible without various members of my team, in particular Praf Arel, who did the simulations along with V1, and the painstaking single channel recordings done by Michael Clausen, who's now back in Denmark, and various other people who support. Thank you.
Stephen, thanks for a great talk. Beautiful work, great interpretation. Questions, please.
When the jet liposome is attached on the glass surface, what interaction are going between the bilayer and the glass surface?
I have no idea. I mean, there's lots of studies simulating that. Both. The actual physical nature of those interactions, I have no idea, I'm afraid.
The seal resistance is over giga ohm or some more than that.
Oh, so the seal yes, we can get. I mean, we have to get very high seal resistances for these type of recordings. Yes.
Maybe a rather basic question, but are there other pathologies that give rise to this membrane stretch other than mechanical? So heat, inflammation, and therefore could this be a more general phenomenon to other ion channels rather than the one you just talked about now that we're just not aware of?
So I think it's a very interesting point actually. I mean, these channels exhibit this polymodal activation. Many different bioactive lipids will modulate their activity. Lots of small molecules in very non-specific ways. And one of the most potent activators of Trek channels is actually Triton X100. But I think particularly this particular subfamily of K2P channels are exquisitely sensitive to their membrane environment, and anything that perturbs that local environment has the potential to modulate their activity. And maybe we should be thinking about these kinds of things for how other channels are modulated. But I think, any mechanosensitive channel is going to be exquisitely sensitive to its membrane environment. There are a lot of drugs that act very non-specifically on these channels, and the pharmacology is quite dirty in many respects.
Okay, so related to your answer, thank you very much for your talk. Great talk. Is there any data or research about the phospholipid composition influences the tract channel activity?
Oh, sorry, what influences it?
Phospholipid.
Whole cell?
Phospholipid.
Yes, I mean, you can get very different levels of activity depending upon the types of phospholipids that you reconstitute them in. I mean, these channels are very potently activated by PIP2. Mm-hmm. There's various other lysolipids, and any conical lipid will activate these channels, but that's probably through changing the mechanical properties and perhaps inducing curvature around the channel. So whereas some other lipids we think maybe are actually binding directly to the protein and influencing their activity, but also I think there's going to be lots of non-specific effects of lipids as well. Mm-hmm.
Could you comment on your molecular dynamic simulation software? Does it require solved crystal structures?
So does it require
solved crystal structures?
You can do this with a homology model, but the caveat is that it's a homology model. So believe what you want. Mm-hmm.